interconversion of UDP glucose and UDP galactose is catalyzed by. So if at one position we have UDP glucose, okay, and we are having UDP galactose. So which enzyme catalyzes this reaction? Glucose to galactose and galactose to glucose. Four options are oxidase, kinase, epimerase and mutase. Glucose to galactose conversion, what is happening here? Glucose and galactose are, so the reaction will be catalyzed by epimer. What about mutase? What does it do? That also do the rearrangement of the molecules. Here the conversion, if mutase, so in glucose and galactose, what we saw that glucose to galactose, here at the same carbon only the rotation got changed of OH and H, that got replaced. But in case of mutase, what is happening? Of this one, so here the change is happening. Suppose there is a these two carbons. Okay, so here is the X group, here is the H group. So after mutase activity, H group and X group get rearranged, but X comes to this carbon and H goes to this carbon. That rearrangement is done by mutase. But if it have been epimerase, the position will never be changed. It is regarding the same carbon, but here the in regard of another carbon, the rearrangement is happening. That is why there is a difference between mutase and epimerase. Oxidase, we know it is performing oxidation. Oxidase performs the oxidation. Yeah, it is basically phosphorylates the molecule with the help of ATP. That is why it is kinase. Okay, performs the phosphorylation, addition of phosphate group, and that phosphate group has been taken with the help of ATP. So then kinase. Otherwise, if the ATP is not involved in the phosphorylation, then it will become phosphorylase. Okay, phosphorylase. Not write properly. Transfer of phosphate group can be performed by both of these enzymes, kinase and phosphorylase. Okay, but here in case of kinase, ATP is involved, but in case of phosphorylase, ATP is not involved. Okay, so here if the equation would have been UDP glucose to UTP glucose, and ATP would have been involved, then it would have been a kinase activity. Okay, and glucose to galactose, we know what are the changes are happening, that is involved in the same carbon, the rearrangement on the same carbon. That is why epimerase is also not involved. Sorry, mutase is also not involved, and oxidation is also not. From glucose to galactose, there is no oxidation is happening. That is why oxidase is also wrong option. Okay, so the correct option will be the epimerase enzyme. Okay. Mutase. Okay, so mutase is, so if there is something like, suppose there is a 2 carbon molecule, right? This is a molecule. Okay, this is one molecule. So the changes, this is the same carbon. Now you can see that at C1 carbon you are having X. Okay, but after mutase activity at C1 carbon it will have H. Another, the composition is not changing here also. The composition is same in this case and in this case. Only rearrangement is happening from one carbon to another carbon. So Now C2, which harbors H, now it harbors X. So the rearrangement is happened like this. Okay. So if that kind of rearrangement is happening, then it is called mutase activity. So for example, in we study in glycolysis, there is a one-step conversion, 3-phosphoenol 3-phosphoglycerate. If I'll say not in glycolysis, if I'll say 3-phosphoglycerate, glycerate, if it looks something like this, 3-carbon molecule. Okay, here is the phosphate group. I'm just denoting it with P. Okay, H, here OH, so this. Okay, if this is 3-phosphoglycerate, after mutase activity, looks something like this. So now phosphate group has shifted from third carbon to second carbon. So from 3-phosphoglycerate, it will become 2-phosphoglycerate. Okay. So this is what the mutase function. So it is performed by glycerate mutase enzyme. Okay. Glycerate mutase. Okay. But in case of epimerase, what we are finding that in the same carbon, the rearrangements are happening. Okay. Like at C1 carbon, if we have OH and H like this situated via rearrangement, rotational rearrangement, it can look like this, H and OH. Change in the rotation in the same carbon, okay? This particular carbon only, okay? But in case of mutase, shift is happening from one carbon to another carbon. That is the mutase. These two are very like direct questions, okay? You just need to know the basic. That's it. You can easily solve them and don't get confused with other enzymes. So you should have an idea exactly what each enzyme performs the function and what is exactly happening in the reaction which is given. So here in this case, you must be knowing what is happening when glucose is converted to galactose. What kind of rearrangement is happening. Okay. And what are the 4 options? How they perform the reaction. My NPTEL reference. You can refer to this lecture to understand more detail about carbohydrates, epimer, and many different aspects of sugars. Thank you.